Okay, this is session 2. We're going to be having some talks focused broadly on addiction. And the first one is going to be from Kay Lara from my lab. And she's going to be talking about genome-wide association study of delayed discounting in heterogeneous stock rats. And take it away, Montana.
Thank you, Abe. Let me put my podium stand up this way. Yeah. Okay, hi everyone. I'm super excited to be here. My name is Montana. I am a new postdoc in Abe's lab, but I'm coming from Matt Mahoney's lab, who is also here. So talking about some GWAS in HS rats. And I wanted to sort of start with the HS rat population because I am the first person to talk out of Abe's lab, and you'll see this slide again and again. But the HS rats are an outbred population that is derived from 8 inbred founder strains, and they have been used extensively in GWAS, as you know. So because we have a high level of both genetic and phenotypic diversity, these animals are a really ideal population to study complex neurobehavioral traits and associate genes with those traits. It's not going to the next slide. Oh, there we go. Okay. Apologies. Okay. So broadly, we measure some variable phenotype in these rats, and then we want to be able to conduct some genetic association with those phenotypes and be able to explain that phenotypic heterogeneity with our genetic association studies. And so today what I'm going to talk to you about is delayed discounting, which is the tendency to prefer some smaller, more immediate reward over a larger delayed reward. So for example, less money now versus more money later. And it's often measured by giving individuals a series of choices between two rewards where that reward size and that delay in time is systematically adjusted. And after all these adjustments, we arrive at an indifference point. And this is where those two options are subjectively considered to be equally desirable and equal in value, even though they have different delays and the magnitude of the reward is also different. And so we can plot those indifference points along the delays of the adjusting trials. And yes, okay. So we're plotting those indifference points along that delay, and what we get are discounting curves. And so these are two examples for individuals, and each point on the curve represents that immediate reward amount or the indifference point that is equivalent to the reward at the specific delay in time. And so really the take-home message here is that a steeper curve means that the individual has discounted that reward at a higher rate. So they have a greater preference for the smaller immediate rewards relative to the larger delayed rewards. And importantly, and why I'm part of this particular session on addiction, is we study delay discounting because there is a wealth of evidence that shows us that individuals with substance use disorders consistently have higher levels of delay discounting. So you could imagine this relationship with delayed discounting and substance use disorders where individuals would prefer a more immediate, though smaller or maybe more short-term reward, and discount or devalue that more future long-term reward to a higher degree. And you know, at this conference, we care about genetics. So obviously, we want to understand the underlying genetics of this behavior because through familial and twin studies, and of course GWAS, we know that there is an established genetic component to delays discounting. And so To do this in model systems, what we're going to use is an adjusting amount procedure. And this work comes out of Suzanne Mitchell's lab at OHSU, who I believe should be here online as well. And so let's take a look at this adjusting amount procedure really quickly. Essentially, this is a neurobehavioral assay. A rat is put in a box and there's lights and levers and a little sucrose receptacle for them. And they're just started out in this box in the initiation phase. And then they're moved on to the choice phase where these two levers are going to be illuminated and the rat is going to make a choice. And so they can press the delay lever, and after some varied amount of delay, they are going to receive 150 microliters of a sucrose solution, or they can choose that immediate lever. And so they're going to be given some variable amount of the sucrose solution, and that's going to vary based on their previous choice, but it will be an immediate reward. So if they press the delay lever, then the next time they press the immediate lever, that size of the immediate reward is going to go up by 10%. And then if they press the immediate reward and then the immediate reward again, the size of that reward is going to go down by 10%. And then there's this timeout phase. And then we do it all over again, right? Or the rat does, not us. So the rat is gonna get up to 60 trials, or about 60 minutes. And so the basic assumption of the adjustment method is that these animals will eventually titrate that adjusting amount until it basically equals the subjective value of that delayed but fixed reward. And so at that indifference point, these animals will switch back and forth between that adjusting and delayed alternative. And so each of these sessions with the 60 trials is done 6 times per these 6 different delays. So 0, 2, 4, 8, 16, 24 seconds. And so for each of those delays at those seconds, we can plot the average indifference point for that animal at that delay. And here I'm just showing you the discounting curves for all the animals in this study. And the black is the average for all the animals. And so you can see kind of similarly at the top, those are the ones with less discounting versus the more steeper curves have higher discounting. And so this slide is in here really to tell you what a great lab I just joined. So essentially what we're doing now is we're taking the phenotypes and then we're going to do genotyping on them and then conduct some genetic association study. So like I said, this delay discounting was done in Suzanne Mitchell's lab. Genotyping, we do low coverage whole genome sequencing and then impute using Stitch, and we get this really, really low error rate. So we've really perfected this. So it's just so amazing. And then through countless association studies that have been conducted in the lab with many, many collaborators, we've really optimized this GWAS pipeline. So thank you to all these people. And so for delay discounting, what we've done is taken the area under the curve for those curves, the discounting curves. And here we detect a genome-wide significant locus on chromosome 20 for AUC. And also for this particular SNP, the heritability estimate was about 0.2. So in line with other HS rat studies for heritability. And so this particular SNP on chromosome 20, it maps to the SLC35F1 gene. And just to take you on a quick dive into what this gene is, it encodes a member of the solute carrier family. So it is a nucleotide sugar transporter. And it is going to mediate the transport of molecules across membranes, particularly in the Golgi and ER. And we also know that it is highly expressed in fetal and adult brain tissues in humans. Other studies have shown high expression, particularly in the soma and dendrites of neurons. And so there was another study that has this like possible involvement in the formation and function of dendritic spines. And so while there's this high neuronal expression and potential role for dendritic spine dynamics, these all point to this brain-related function, but obviously we don't fully appreciate the molecular mechanisms by which this gene is participating in neuropsychiatric behaviors. That remains to be elucidated. Oh, sorry. Yeah. And then importantly, to sort of bring this back to the human aspect, this gene has been related to certain clinical presentations. In one case report study of 6 unrelated pediatric epilepsy patients, they all had microdeletions in this region. If you know anything about my previous research, this interested me because I worked on epilepsy before. And similarly, there was a mutation found in a patient with something called Rett syndrome-like presentation. So Rett syndrome is a disorder that involves like motor, cognitive, and behavioral impairments. And then finally, for GWAS is for other related neuropsychiatric disorders and neuropsychopathologies. This gene has sort of been identified not for, it didn't reach significance for genome-wide significance, but it neared significance for bipolar disorder, ADHD, and schizophrenia. It was significant, however, for educational attainment, which, so lifetime outcomes such as educational attainment have actually been negatively associated with delayed discounting. So individuals with steeper delay discounting have lower educational attainment rates. So there has been a mouse model of this with the Knockout Mouse Project, but they were unable to recapitulate any sort of neuro-related trait that is seen in humans with that particular mutation. But obviously they didn't do delayed discounting or some similar behavioral construct, so it remains to be seen. Also, it's on a Black 6 background, so lots of points we could make about that. It was unfortunate, but I'm not discouraged. And so in conclusion, modeling of delayed discounting in rats definitely has disease relevance, as we know. And the HS rats are a great population for this because they do have variable phenotypic presentations. The GWAS that we conducted resulted in a QTL on chromosome 20, mapping to the SLC35F1 gene. And this gene has neurodevelopmental implications and should definitely be explored further. Thank you to the Palmer Lab, the Mitchell Lab, the HS rat. Sorry, the screen just changed. Leo Solberg Woods at Wake Forest. This is a picture that I'm not in, but I will be in the next picture for the next CTC meeting. So thank you very much, and I'll take any questions.